The data available to human beings has exploded. Data mining and modeling techniques are swamped by the amount and the dimensions of data. Facial recognition is one such area with large amounts of data and many factors to each image. Factor analysis can be used to reduce the amount of dimensions to help keep the computational burden down when using data sets with a large amount of dimensions. In this experiment, PCA is used to reduce the dimensions in a 96 by 96 image in order to make predictions about 15 facial key points. These key points can then be used in further facial recognition algorithms. What you see here are the eigenfaces. That is the result of running PCA on our images and visualizing the features that are extracted by each principal component. With PCA, the question is how many components to use. Here we see an attempt to find the right number of components to accurately predict a few key points. This early study showed that varying the number of PCs helps converge to a better solution, but only up to a point at which the error gets worse. So in order to predict all of the facial key points, 30 X and Y points in all, how do we choose the optimum number of principal components? And does the same amount of principal components predict best for each key point? This question can be answered by searching for the best solution over the solution space. But as the number of key points goes up and the number of PCs is still fairly high, this becomes a hard combinatorial problem. One technique to handle this search is called genetic algorithms. These are used to encode a gene, no, not that gene, that stands in for the combination of the differencing number of PCs used to predict each key point. Imagine that each position on the gene represents the number of PCs for that key point. So thus, we have a gene that is 30 positions long, and which, in our case, can have up to 100 different PCs used to generate the prediction for that key point. The genes produced are then used in linear models to individually predict each key point and are graded against the known values, and the whole gene is then graded against other genes. The best genes are retained to live another generation, as in natural selection, and these genes are then subject to mutation and crossover to continue the search of the full solution space. The most interesting result in this experiment is that different positions on the gene used to predict different key points seem to require very different numbers of components to best make a linear predictor model. The winning gene in our final experiment ranges from using 15 up to 35 different principal components as input into their respective linear models. So, in some areas of the image, a small number of PCs served to predict the point well, and in others, a larger number of PCs were used to predict a key point.